Hello everyone, the bio professor is back, and today we are back with a very important topic of 11th NEET syllabus, the plant physiology, and within plant physiology we'll be discussing a very important subunit today. Which is that one? Let's see after this small introductory part itself. So you all are clear now that we are going to discuss about glycolysis today, and we are going to discuss as much detail as possible in glycolysis. We will also see that what are the minor points as well as what are the major points which are required to be kept noticed always in our mind while we will be writing the questions or while we will be taking the questions on glycolysis topic itself. Okay, so let's start off the topic. It's, I would say, a very short topic as compared to that of the Krebs cycle itself. So first of all, as I always used to say, biology or the science, it is a topic which is actually the meaning of all the concept, it is hidden within that of the word itself. So if I'm saying glycolysis, the word, if I'm splitting this two word, we can get it as glyco plus lysis itself, in which glyco word that stands for carbohydrates itself, whereas if I'm saying about lysis, this stands for what? This stands for that of breaking down. So glyco stands for what? Glyco stands for that of carbohydrate itself, whereas lysis, it stands for breaking down in that case. Okay, so let's consider that if I am taking a cell. Let's consider that if I am taking a cell itself like this, let's say. So if I would like to brief the different cellular respiration steps which are occurring, how can I brief those things? Like if this be a cell, okay, and within this cell itself, let's consider that this be the mitochondria, which is having its own invagination. We know mitochondria can easily be identified with the help of all these invaginations itself. That is the cristae which are remaining present like this. Okay, so now if we are saying, if we are briefing all the steps within that of the cellular respiratory pathway itself, first of all, what will happen? The C6, H12 and O6, this carbohydrate, it is first of all required to be broken apart in the cytoplasmic area only. So first of all, the carbohydrate will be broken apart into a small intermediate, a small 3-carbon intermediate, which we are representing it as the pyruvic acid itself. And that breaking down of that carbohydrate, it will be taking place within that of the region which we are representing it as that of the cytoplasm itself. That is the region which we are representing it as that of the cytoplasm itself. So first of all, as soon as this carbohydrate is getting broken apart into that of the pyruvic acid, after that what will happen? Now there will be furthermore intermediate reactions and other reactions itself, by which all the intermediates from the pyruvic acid, it will get further entered within that of the mitochondrial matrix, and via getting entered within that of the mitochondrial matrix, finally it will result to the release of that of a very important molecule which we are representing it as that of the ATP itself. So by this only it is clear, I hope so, that the topic of our discussion, that is the glycolysis, if we are saying the location of that of glycolysis, it is actually what? The location of glycolysis, it is actually the cytoplasm itself. Now in your all mind, it might flash that why, although all the steps, other steps like the Krebs cycle, like the terminal oxidation and all other steps, well, it is occurring within that of the mitochondria, why this step, that is the glycolysis, it is occurring in the cytoplasmic region or the protoplasmic region itself? The major reason for the same is that in order to break down carbohydrate, it is always required a higher number of enzyme, as well as it is always requiring the presence of that of a larger surface area. So breaking down of that of carbohydrates itself, and those enzymes which are involved in breaking down of that of the carbohydrate, they are mainly found or located in that of the cytoplasmic or the protoplasmic region, and because of which this step, it is occurring within that of the cytoplasm itself. Moreover, the second point is also there, that the cytoplasm or the protoplasm, it is providing a bigger surface area to that of the breaking down, or to that of that process itself in that case. Okay, so if we are saying, we can say that this is a process which is which we can also refer it as that of an anaerobic process also. This is a process which we can represent it as that of an anaerobic process. Why we are representing it as an anaerobic process? Because what is happening, this is a process which is occurring in that of the cytoplasm or the protoplasmic region. So this process, it never consumes oxygen, and when it is not consuming oxygen, we know that all of those steps, all of those processes which are not consuming oxygen, we are always representing them as the anaerobic process itself. Okay, so if we are seeing some important points, some important detailed point regarding that of glycolysis, let's see all those important brief points. Let's how we can brief these points itself. So first of all, if I'm saying glycolysis, this is a part of that of certain important pathways, or we can say that these pathway, they are also named as that of EMP pathway itself. Now, why we are naming it as EMP pathway, we know because there were three scientists involved in discovering these glycolysis and breaking down of that of these respiratory substrates itself. They are named as Emden, Meyerhoff, and Pernas. And based on their name, they had said that generally the carbohydrates or the these major respiratory substrates, they used to follow this special metabolic pathway in order to get broken apart. In that case, so they say that breaking down of glucose, C6H12O6, is also a part of this pathway itself only. So we are representing them as that of the EMP pathway itself. So as we had seen, let's brief all the points itself. As we had seen that this step, it is occurring. First of all, do remember, in any of the steps, in any of the processes, whenever you're studying any process, let's say whether it's central dogma, transcription, translation, and replication, if you want to love those processes, you have to start from that of the scratch, and you have to go into that of the depth itself. Okay, so when you are going to depth, at per your limit, don't research over the topics, because NEET is never wanting researchable topics from your end. NEET is wanting you to be limited, strict to that of your own point of discussion, whatever it has been asked or whatever it has been mentioned within that of the NCERT level itself. We have to explore all those points, but to a certain extent. So whenever you are studying any kind of topic, let's say about replication, let's say about transcription, translation, respiration, photosynthesis, all this thing, try to start from that of the scratch itself. Just an example I'm saying, suppose if you're studying about replication, try to analyze the topic that replication occurs in which stage of the cell division, because we know, first of all, the cell will get divided, and prior to that of the cell division, all these processes are required to be taken place. So in which sub step the replication or the central dogma steps will be taking place? That is the scratch level question. That is a basic level question. So if you are starting to analyze all these points from that of the basic level questions only, automatically you will get more amount of passion, more amount of love for that of your subject itself. In the same manner, we are discussing over here also. Location, as we had already discussed priorly also, that cytoplasm, it is occurring within that of the within that of the cytoplasm, I'm sorry, within that of the cytoplasm, the EMP pathway or the glycolysis, it is occurring in eukaryotic cells, as well as if I'm saying in that of the prokaryotic cell itself, the glycolysis, this step, it can occur within that of the protoplasm itself. Because why I'm saying protoplasm, you all know, because in prokaryotic cells, they do not possess any nucleus or any nucleoplasm kind of thing itself. So we can say that in general, we can say that within that of the prokaryotic cell, it is occurring within that of their protoplasm itself. Okay, so as because this step, it is never involving the consumption of that of the oxygen. The same point, let me write once more over here so that these points you can recap again and again. It is an anaerobic pathway. Moreover, I would like to say that this is, we can mention it also like this, that this is also a common step. This is also a common step in between that of what? This is also a common step in between that of the aerobic respiration and an anaerobic respiration. Because in anaerobic respiration, they will not be involving the Krebs cycle and other steps, but in aerobic respiration, it will be involving. So glycolysis is only one step which is common in between that of aerobic as well as anaerobic reaction itself in that case, or respiration itself. Okay. So we can say that this is a pathway or this is a reaction which can be referred as that of a metabolic pathway. Obviously, it will be a metabolic pathway because energy involvement is occurring over here. So this will be the metabolic pathway itself. Okay. And as this is a metabolic pathway, so obviously there will be the involvement of that of the enzyme itself. So enzymatic breakdown of the substrate will get occurred within that of this step itself. Enzymatic breakdown will get occurred within that of this pathway itself. Okay, so when I'm saying about that of the enzymatic breakdown and everything, the enzymes, they will be working over that of, or the enzymes, they will be working over that of, we can say, the substrates itself. They will be breaking down those substrates and they will be like emitting kinds of that of, we can say, the ATP and all the whatever the required further intermediate components are there, that will be released in that case. Okay. So a very important point which we need to know, that what or how many types of reactions, how many types of sub-reactions, I would say, or intermediate reactions that I would say can get occurred in glycolysis pathway. How many types of sub-reactions or intermediate reactions that can get occurred in glycolysis pathway itself. So let's see how many types of reactions. If I'm saying, let's brief these reactions itself. The first type of reaction, the very important type of reaction, is always the phosphorylation reaction. And along with that of phosphorylation itself, it will also involve the reaction which we are representing it as that of the dephosphorylation reaction itself. Okay, so it will be involving the phosphorylation as well as it will be involving the dephosphorylation reaction itself in that case. Okay, so if I'm saying about this thing now, phosphorylation means actually what? Phosphorylation means actually it is the addition of that of the phosphate group. We can say that it will be involving the addition of that of the phosphate group to certain intermediate components, and based on all those addition, there will be the like formation of that of phosphate rich components itself. Those steps we are representing it as that of the phosphorylation itself. Now in this condition, I would just like to brief a very important point. This is not only for respiration part, this can be, you can apply it for respiration, this you can apply it for that of photosynthesis also in this case itself. So phosphorylation and its types. If we are saying the different types of that of the phosphorylation, so what are the, or what can be the different types of phosphorylation steps that can get involved, or what are the different types of phosphorylation that are remaining present when we are discussing about that of the metabolic pathway itself. We can say there can be three important type of phosphorylation step itself. Okay, how? The first type of the phosphorylation step, it will be involved in that of photosynthesis, obviously, and that kind of the phosphorylation step, we are representing it as that of the photophosphorylation itself. Now, what is this photophosphorylation? Actually, photophosphorylation, that stands for what? It is the phosphorylation or formation of a phosphate-rich component under the presence of that of the light. So when we are discussing about all these things like light, involvement, and all these things, certainly this step will be included within that of the photosynthesis process itself. Okay. Next, further, after photophosphorylation, we'll be having one more step, which we will be representing it as that of the substrate level phosphorylation itself. So what is this substrate level phosphorylation? Substrate level phosphorylation means it will be like the enzymes will be acting over that of the substrate, and when the enzymes are acting over that of the substrate itself, they will be breaking down those substrates and they will be leading to the release of that of the ATP and other molecules itself. So those things we are representing it as that of the substrate level phosphorylation itself. Okay, and the third step of that of the phosphorylation, we are representing it as that of the oxidative phosphorylation itself. We are representing it as that of the oxidative phosphorylation itself. Now, what is this oxidative phosphorylation? Why I am saying oxidative? Because there will be multiple reducing kind of substances which can get involved. Reducing substances means the substances through which like protons and these things will remain added, like you can say NADH2, NADPH, like FADH2, all these components. And when these components will be like broken apart with the help of that of the enzymes itself by the oxidation process and everything, they will be leading to the release of that of, or they will be leading to the release of that of the ATP itself, or the phosphate rich component. So because of that reason, we are representing it as that of the oxidative phosphorylation itself, and thus we are having three major types of phosphorylation, we can say. And in these three major kind of phosphorylation, only photophosphorylation is the special step which is occurring within that of the photosynthesis process itself. Substrate level as well as oxidative level phosphorylation, it will be occurring where? It will be occurring as a common step. It can occur in photosynthesis or in respiration also, in these things. Okay. Further, if we are saying, we can say that the cellular glycolysis will be a step which will be including both reversible. This will be a step which will be including both reversible as well as irreversible reactions. This will be a step which will be including both reversible as well as irreversible reactions. And there will be, if I'm saying total number of steps that will remain involved over here, it will be always 10. So now, when we are saying about that of the 10 steps, out of these 10 steps, we can divide these 10 steps into two forms. We can divide these 10 steps into that of the two forms itself. How we can divide them into two forms? As because in the first 5 steps, ATP can be seen to be consumed. So we can say the first 5 step as that of ATP consumption step. Whereas in the rest of the other 5 steps, the ATP formation will get occurred. So you can also say that in the first step, as in the first 5 steps, as because ATP is getting consumed, so certainly in the rest of that of the 5 steps, ATP is been formed, so the level can get balanced itself. And along with that also, we can say that some more amount of ATP will be also getting formed, which will be the net product of that of the glycolysis itself in that case. Okay, so we can say enzymes are involved in all these steps. Enzymes will be involved in all these steps in cell, whether it is the ATP consumption step or whether it is the ATP formation steps itself. In all of the steps, enzymes will be the major part in that case. Okay, fine. So if I am saying about the broad steps which are involved in that of the glycolysis. Okay, so what is happening? First of all, we know here the respiratory substrate is there, that the respiratory substrate will be broken apart, certainly. So we can say that glucose, that is the respiratory substrate over here, that is the C6H12O6, it is getting formed, or it is present over here itself. We are gaining it from that of the nutrient part itself. Now this glucose, it will be converted, there will be the addition of that of certain phosphate ion. We can say that within that of these glucose itself, there will be the addition of certain phosphate ion at the 6th carbon atom of that of the glucose, and it will lead to the formation of that of a component which we are representing it as that of the glucose 6 phosphate component itself. So we can say that if I'm saying about that of the glucose part, we can say the glucose is there, enzymes are working over that of the glucose as that of the respiratory substrate. So when enzymes are working over that of the glucose as that of the respiratory substrate, the what will happen, even a phosphate molecule, a phosphate ion will be added with that of the 6th carbon atom of that of the glucose, and it will be leading to the conversion of glucose to that of the glucose 6 phosphate itself only. Okay. Now, as soon as the glucose 6 phosphate is getting formed, certain specialized isomerizing kind of enzyme will be working over that of these glucose 6 phosphate itself. So when it is an isomerization kind of reaction, it has to be the reversible kind of reaction itself. So these isomerizing enzyme will be acting over that of this glucose 6 phosphate, and this will be leading to the formation of that of the component which we are representing it as that of the fructose 6 phosphate itself. This will be leading to the formation of that of the component which will be leading to that of the, which will be reading it as that of the fructose 6 phosphate itself. Okay, so now what will happen? This was an isomerization reaction. Now plainly we can say that this fructose 6 phosphate, again over this fructose 6 phosphate, at its first carbon atom, there will be the addition of that of an inorganic phosphate ion. Over this fructose 6 phosphate, at its first carbon atom, there will be the addition of that of an phosphate ion, inorganic phosphate ion, and that addition of inorganic phosphate ion, it will be leading the conversion of fructose 6 phosphate into a very important component, into that of a very important component. I'm just writing F, that stands for fructose, very important component. We can represent that important component as that of fructose 1,6 biphosphate. You can also say diphosphate also, fructose 1,6 diphosphate or fructose 1,6 biphosphate itself. So it will be leading to the formation of that of fructose 1,6 biphosphate itself in that case. Okay, so this we can say that substrate which whatever it is getting formed, okay, this is a very important one, because this substance, it is also known as an intermediate molecule within that of the whole glycolysis itself. And the fructose 1,6 biphosphate, formation of that of the fructose 1,6 biphosphate, it always the whole step, it is always dependent on that of the formation of that of the fructose 1,6 biphosphate itself. That means if the cell is in need of that of ATP, certainly fructose 1,6 biphosphate will get formed, and as soon as certainly fructose 1,6 biphosphate it is getting formed, so automatically what will happen, the process will be proceeding towards that of the forward direction itself. Now, what is the importance of that of this substance? Let's see. Fructose 1,6 diphosphate. Fructose 1,6 diphosphate, it will be showing splitting into two important components. The first important component, we are representing it as that of the phosphoglyceraldehyde. In short, many students used to say it as also pagal. By jokes apart, I would say this is the phosphoglyceraldehyde, and this fructose 1,6-diphosphate will be again splitted into one more molecule. We can represent it as that of the dihydroxyacetone phosphate, or DHAP. So now what was happening? Let's see about these points. Glucose, it was of 6-carbon component, and one molecule of glucose was involved over here. Here also one molecule of glucose 6-phosphate was involved, and 6-carbon component. Along with that, fructose 6-phosphate, again one molecule and a 6-carbon component. Fructose 1,6-diphosphate, it is also a 6-carbon component, and suddenly this fructose 1,6-diphosphate, it will be broken apart into two components, and these two components, they will be of 3-carbon compound in nature. These will be of what? These will be of 3-carbon component in nature. So now you can involve, there is no, now you can relate that why there is no involvement of the other oxygen, because like there is no oxidation process over here. It's like a splitting which is getting occurred over here itself, and simply the molecule, when it is getting splitted apart, it will be leading to the formation of that of the 3-carbon components itself in that case. Okay, now what will happen? We got one molecule of PGL, we got one molecule of DHAP, that is dihydroxyacetone phosphate. So now these two are isomers of each other, and as soon as DHAP will get formed, DHAP will be reversed into that of the PGAL itself. So priorly there were one PGAL. Now what will happen? We used to say it as priorly there were only one Pagal. Now after the conversion of DHAP to this component, we are having two Pagal. Again, jokes apart, we are having two molecules of phosphoglyceraldehyde itself. Okay, so now the major game starts from that of this phosphoglyceraldehyde only. Okay, and this will be proceeding towards the further direction. So what will happen? If I'm saying about that of this phosphoglyceraldehyde, now with this phosphoglyceraldehyde, over this phosphoglyceraldehyde, there will be again like a phosphorylation kind of the other step, as well as dehydrogenation kind of step itself. So now dehydrogenation over what it will be happening? There will be a molecule which we are representing it as the phosphoric acid, H3PO4. So now what will happen? This H3PO4 will get dehydrogenated. Now, as soon as the H3PO4 is getting dehydrogenated over here, immediately what will happen? It will be showing the release of that of the phosphate ion, as well as it will be showing the release of that of the proton ions itself. And there in this vicinity, whatever the enzyme is remaining present, along with that enzyme, there will be the presence of that of NAD plus component, nicotinamide dinucleotide itself. So that NAD plus component will be accepting this proton ion, and it will be leading to the formation of that of NADH2 itself. Okay, so and this inorganic phosphate, now this inorganic phosphate, it will be like accepted by that of phosphoglyceraldehyde itself, and now this will be leading to the formation of that of the component which we can represent it as that of 1,3-biphosphogyceraldehyde itself. So what will happen? I'm repeating once more. First of all, fructose 1,6-diphosphate got formed. As soon as fructose 1,6-diphosphate got formed, immediately what had happened? Immediately the fructose 1,6-diphosphate will be broken apart. And as soon as the fructose 1,6-diphosphate is getting broken apart, immediately what will happen? It will be releasing 2 3-carbon molecules as phosphoglyceraldehyde and dihydroxyacetone phosphate. Now both are isomers of each other. This dihydroxyacetone phosphate, as soon as it is getting formed, immediately it will get converted into that of the PGAL, that is phosphoglyceraldehyde itself. And as soon as it is getting converted into that one, we were priorly having one molecule of phosphoglyceraldehyde. Now we will be having two molecules of phosphoglyceraldehyde, and whatever the steps now it will be following, all those will be having the 2 molecule substances only. Prior to that of in ATP consumption step, it was all the substrates were of 1 molecule only. In all these steps, it will be of 2 molecules. Okay. So Now this 1,3-biphosphoglyceraldehyde, it got formed by this dehydrogenation process, and it resulted to the formation of that of this first assimilatory part, that is the NADH2 itself. Now this 1,3-biphosphoglyceraldehyde, it will be showing the dephosphorylation, the first dephosphorylation step, okay, in which the phosphate group, inorganic phosphate group, will get left from here itself. As soon as the inorganic phosphate it is getting left from here, immediately in this vicinity there is the presence of that of the ADP, and immediately what will happen, the ADP will be combining with that of the inorganic phosphate ion, and this will be leading to the formation of that of the ATP itself. So ADP will get combined with that of the inorganic phosphate, this will be leading to that of the ATP itself in that case. So 1,3-biphosphoglyceraldehyde will like now be broken apart, and as soon as 1,3-biphosphoglyceraldehyde it is getting broken apart, it will be leading to the formation of that of a component which we are representing it as that of the 3-PGA or 3-phosphoglyceric acid. Okay, now as soon as this 3-phosphoglyceric acid is getting formed, immediately this 3-phosphoglyceric acid, now it will get converted into that of, like, there will be a kind of structural isomerism, you can say, it will be happening over here, and this 3-phosphoglyceric acid, suddenly it will get converted into that of a component which will be representing it as that of the 2-phosphoglyceric acid itself. This will be getting converted into that of a component. We will be representing it as that of the 2-phosphoglyceric acid itself. Okay. So as soon as this 2-phosphoglyceric acid is getting formed, now this 2-phosphoglyceric acid, it will be whatever the enzyme will be acting over that of this 2-phosphoglyceric acid itself, what it will be doing, it will be showing a dehydrogenation from that of this 2-phosphoglyceric acid, and it will be showing the release of that of water molecules. Okay, and as soon as the water molecule will get released over from here, what will happen, this 2-PGA, now it will get converted into that of molecule which we are representing it as that of phosphoenol pyruvic acid, phosphoenol pyruvic acid itself. Okay, this is the, I would say, second last component and a very important component itself. So phosphoenol pyruvic acid, now this will be showing the typical dephosphorylation step, and as soon as this is showing the dephosphorylation step itself, finally this phosphate group and enol group will get released from here, will be leaving the component which will be representing it as that of the pyruvic acid itself, that is the PA, the PA, which will be of 3-carbon component. So you can understand over here, PGAL was 3-carbon component, this was a 3-carbon component, this was 3-carbon, 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 and finally we got the product, it is of 3-carbon component. So how many molecules of pyruvic acid will get formed? 1 glucose without any oxidation, without any decarboxylation, I would say. There is no decarboxylation step which is getting involved over here. So without any decarboxylation, the 1 molecule of glucose, that is 6-carbon atom, okay, it will get broken apart into that of 2 molecules of that of intermediate component, 3-carbon intermediate component, which we are representing it as that of the pyruvic acid itself. I'm writing 2 comma, doesn't mean that it is like a molecular structure. It means 2 molecule of that of pyruvic acid itself only. Okay. So this last step will show, this is the first step where the ATP is getting generated, and this is the last step where is the second ATP will get generated itself. So this is the step. Now, these are the steps which are mentioned everywhere. Now we have to know that what are the important points which are getting involved in that of the glycolysis. There can be many important points. Like, first of all, important point, how many molecules of that of like ATP in this payoff phase, I would say. Payoff means where the ATP has been returned. Actually in gross it is getting formed. So you can see all these are involved as 2 molecules itself. So when these are all involved as that of 2 molecules, so obviously here will be 2 molecules of ATP will get involved. Here also there will be 2 molecules of ATP that will get involved over here itself. The total, if I'm saying the total substrate level ATP, whatever it will be produced, it will be 4 molecules of ATP itself. Now we have to see over here. Here, actually what is happening, first of all, one molecule of glucose, it was getting converted into that of glucose 6-phosphate. So when it is getting converted over here to glucose 6-phosphate, obviously it is showing the consumption of that of one molecule of ATP. So what is happening over here, one molecule of ATP, it will be broken apart, and this will be showing the release of that of ADP and the inorganic phosphatidine. And whatever the ADP and the inorganic phosphatidine will get formed, that inorganic phosphatidine will be accepted by that of the glucose at its 6th carbon atom and will result to the formation of that of glucose 6-phosphate molecule itself. Okay, so this was the first step where this ATP was being consumed. Now here you can observe this step, fructose 6-phosphate. Now this fructose 6-phosphate here also, what is happening? ATP is involved over here. Now this ATP, it is showing, we can say that ADP plus PI. So ATP, it is getting dephosphorylated over here. It is showing the release of that of the inorganic phosphatidine itself. And that inorganic phosphatidine will be accepted by that of this fructose 6-phosphate at its first carbon atom. And it will be leading to the formation of that of fructose 1,6-biphosphate itself. So in general here, 2 ATP were being consumed. Here 4 ATP were being generated. So when there are 2 ATP are being consumed, so out of 4, 2 ATP, so 2 substrate level ATP, again I'm repeating, 2 substrate level phosphorylated ATP will be returned back to that of this cytoplasm itself. So net substrate level ATP that will be produced, it is always 2 molecules itself. And from NADH2 by terminal oxidation, we know that 1 molecule of NADH2 always releases 3 molecules of ATP. So here there is the formation of that of 2 molecules of NADH2 itself. So when there is a molecule, 2 molecules of NADH2 will get formed. So 6 ATP molecules will be getting generated over here. So altogether, if I'm saying from that of 2 PGAL, just remember my point, from that of 2 PGAL in the whole glycolysis pathway itself, there will be the production of that of 2 molecules of NADH2 and 4 molecules of substrate level ATP itself. 2 molecules of NADH2 will always be giving me 6 ATP by terminal oxidation, and 4 ATP it is always already there by substrate level phosphorylation. So in gross, within this glycolysis, it will be leading to the formation of that of 10 molecules of ATP itself. But out of 10 molecules of ATP, it has to return 2 molecules of ATP to that of the the cytoplasm itself. So if I am saying about that of the product which is involved within that of the glycolysis itself, the product in sense, what are the products? Net ATP, I would say, that will be getting involved. So let's see the products which are getting involved. So there will be the always 2 molecule of pyruvic acid. From 2 PGA, it will always release 2 molecules of pyruvic acid. Let's say about net productivity, I would say 2 substrate level ATPs will get produced and 2 NADH2 will get produced itself. So do remember here I am saying as per 2 ATP molecule stands for, I'm mentioning about that of the substrate level ATP itself. Okay, so if in question, if in NEET they are asking you about that how many ATP molecules will get produced from 1 PGL, just halve this one. Okay, whatever you're getting, just halve this one. 1 PGL, how many molecules of ATP will be getting formed in all of the steps itself? Just halve whatever the part that you're getting in this thing. Just do remember all these tricks of the bio professor. Okay, you will be succeeding a lot. All those important tricks, they are very important in any chapters, whatever we will be discussing from now onwards. Okay, very fine. Now we have to know one more important thing. We have to know about that of the enzymes. Students used to say, me, sir, there are lots of enzymes, we have to mug it up. But I always used to say that there is no need of mugging. If you are understanding the types of the reactions which are getting involved, and if you are just making certain tricks out, there is no need of mugging. Just practice it out and you will be able to remember all the enzymes. Let me give you an example. Let me explain my point. Now here, 1 glucose, it is showing the acceptance of that of 1 phosphate ion itself. So one glucose, when it is showing the acceptance of that of one phosphatation, and there is a dephosphorylation step is happening. Do remember, you can remind this step like this: key whenever in like plant physiological or metabolical processes, whenever there will be like the dephosphorylation or phosphorylation kind of steps will be getting involved, that will be always a kind of like kination kind of reaction. You can remember like this the enzyme name. Okay, and whatever the enzyme that will be involved in that step, we will be referring that enzyme as that of the kinase itself. So the kinase enzyme here is working over that of the 6 carbon atom, that is the glucose itself. So obviously we can name the enzyme which is involved over in this first step as that of the hexokinase itself. Okay, very easy. Just try to identify the reaction. As soon as you're identifying the reaction, then after that, the nature of that of the substrate molecule over which the enzyme is acting or will be acting, just add that nature itself. You will be getting the answer. Okay, one more example I'm giving you. Glucose 6 phosphate to fructose 6 phosphate. So glucose 6 phosphate, glucose 6 phosphate to fructose 6 phosphate, as I said earlier only, that this is actually an isomerization reaction. So when this is an isomerization reaction, certainly there will be the involvement of the isomerase itself. So easily you can name it as glucoisomerase, hexoisomerase, you can say like that. Okay. Fructose 6 phosphate to fructose 1,6 biphosphate. Okay, now this one again here, like for the dephosphorylation, it is getting happened, so kinase will be there. So fructohexokinase or fructokinase also you can say. Now this is a very important reaction, the fructose 1,6 biphosphate. Now here this is leading to the formation of that of what? This is leading to the formation of that of I would say from here, from fructose 6 phosphate till that of fructose 1,6 biphosphate itself. That enzyme which is getting involved over here, we are naming it as that of the fructokinase. You can also say it as that of the phosphofructokinase itself. And as I said you earlier also, that fructose 1,6 biphosphate, it is actually the most intermediate component involved in that of the glycolysis. Now why is it so? Because this enzyme, that phosphofructokinase, this enzyme, it is like a very important intermediate enzyme itself. It is always allosterically inhibited by that of the presence of ATP in the cytoplasm as well as that of the citrate itself. Now, what is this allosterically inhibited? That stands for what? If the cytoplasm, it is already having a large amount of a large concentration of ATP, automatically phosphofructokinase will stop its function and the glycolysis process will not be proceeding. Immediate basis, on the contrary, I would say, if there is a scarcity of that of the ATP, automatically the phosphofructokinase will be converting this fructose 6 phosphate into that of the fructose 1,6 phosphate biphosphate, and the process will be split, like forwarding in the forward direction itself. So for that reason, this enzyme, in short, we used to also represent this enzyme as PFK, phosphofructokinase. This phosphofructokinase is a very important enzyme that you have to remember. Okay, now this fructose 1,6 biphosphate, it is getting splitted apart. This is a condensation kind of reaction in which you can say that an aldehydic as well as a ketonic group of compound is getting formed, and this condensation reaction we will be like this is the enzyme which will be involved over here. That enzyme we can name it as that of aldolase, because this is a kind of aldol kind of condensation which is getting occurred. You will be studying in chemistry, and based on that we are representing it as the aldolase kind of that of enzyme itself. And in order to know all these like aldol condensations and everything, you will be certainly studying within that of organic chemistry itself as such, and you will be coming to know about all details in this thing. Okay, so this PGAL, okay, now what will happen? This PGAL, the phosphoglyceraldehyde, this phosphoglyceraldehyde will be showing the dehydrogenation process. Now, dehydrogenation process over here, what will happen? The phosphoric acid is getting broken apart. So there is a dehydrogenation is happening, as because the main reaction which is happening over here is a dehydrogenation. So we'll be naming the enzyme over here as phosphoglucodehydrogenase, or like that. Dehydrogenase will be the major category of enzyme that will be involved over here itself. Again, here this is a kinase because there is the formation of that of the ATP. As I said you priorly also, that whenever there will be the phosphorylation or dephosphorylation, you can say that there will be the involvement of the kinase enzyme. So this is a kinase enzyme reaction. Now, what is happening? From 3-PGA to 2-PGA, what is happening? This is a sudden change. It is getting happen, and both these are isomers of each other. It's a kind of structural isomerism, but in a sudden manner itself. So it's a kind of mutation. So the enzyme which will be involved over here, you can say it as phosphoglycerolmutase, or simply you can refer it as the mutase also in this case. Okay, 2-PGA to PEPA. Here one important group is being added, this E group, that is the enol group is being added over here. So this enol group is being added by that of the enolase enzyme itself. So when enol group is added over in this then this component itself, the enolase enzyme will be doing this addition of this enol group in phosphoenol pyruvic acid itself. Finally, again, there will be a phosphorylation reaction that will get involved over here itself, where the kinase enzyme will get involved. And which kinase? Again, that pyruvate kinase enzyme in that case. So it's very easy. It's very easy. Just you have to find out the trick. As I always used to say, you have to find out that smart trick for you. And as soon as you're getting that smart trick, automatically you will be like able to sort out any kind of issues in your life, whether it's this study related issue or whether it's kind of any kind of issue itself. Okay, now one more question, the important questions which we are like focusing on. Okay, the reversible and irreversible reaction. So in this step, do remember, in glycolysis, among all these 10 steps, there will be 7 steps which will be reversible. Most of the steps are reversible ones. 7 steps are reversible and 3 steps are irreversible. So in order to like remember which are reversible and irreversible, it is very easier to remind of that of the irreversible steps because the number is less. So what can be the irreversible step? Let's apply again some logic over here. If the last step, if it is reversible, then we will never get the pyruvic acid. Whenever it is getting formed, again it will be going back, again the process will be like repeating. So the last step, it is always an irreversible one. The intermediate step, the formation of this fructose 1,6 biphosphate, it is an irreversible step. And this first step, glucose to glucose 6 phosphate, obviously the first step has to be irreversible one. If it is reversible, the process will not be proceeding faster. So there are 7 steps which are reversible and 3 steps which are irreversible that are getting involved. So it's very easy. Just you have to remind these products. See, always you have to remind that in NEET there is very less chance that they will be just lifting all the complete like flowchart and they will be asking you the questions from that one. They generally, the people who used to set the paper in NEET, they generally used to ask questions from basic point, like which enzyme is involved in the conversion of glucose to glucose 6 phosphate. Which enzyme is involved in that of like you can say like the conversion of fructose 6 phosphate to fructose 1,6 biphosphate. Why we are referring phosphofructokinase or PFK as the most important intermediate enzyme? Why it is allosterically inhibited? All these kind of the question. Again, one more important question, as I said you, that if from 2 molecules of PGAL, the total whatever the amount of ATP is getting formed, then how many will be getting formed from 1 PGL? Simple thing, just you have to simply calculate the total amount and you can get the complete result part itself in that case. Okay, so that was about today's topic discussion. In that case, okay, we had discussed about the glycolysis part. Very soon, we're going to come with some of the important questions related with that of glycolysis, as well as the oxidative decarboxylation and all these kind of part itself. Stay tuned. Please subscribe, as well as don't forget to hit the bell button in order to like get more attractive videos. Do subscribe. You will be benefited. We all will be benefited. Thank you very much.